Well, good morning, Suffolk Punch America. I hope you can hear me and see me. I'd like to welcome you to this side of the pond from Newmarket in Suffolk. And what you see here on the screen is the town sign to Newmarket, which is where I live and work. Actually, I live a little nearby in a village called Wicken, near the cathedral town and birth of Ely, Cromwell's birthplace. On the Newmarket sign, you can see the head of King Charles II, who was one of the great patrons of Newmarket. Newmarket probably being the equivalent of the Las Vegas of the 17th century. Newmarket is extremely well known as the home of thoroughbred racing. And you can see from the slide, we have both horses in training here, a large population of flat thoroughbred racehorses for flat racing, and in the surrounding areas, a large breeding population of thoroughbreds. Historically, the region has bred diverse breeds, and of course, is the heart of East Anglia, which is the home of the Suffolk Punch. Let me see if I can get the next slide up. Where are we going? Next. I hope it'll work. Let me try this. Go through to here. Why not? It worked earlier on, but I'm trying to get to the next slide. In a second. Right, where am I? Well, I'm the blue dot in the top right-hand corner, and you are the red dot over there on the other side of the pond. I'm also the blue dot right here, north of London. We're 60 miles north of London, a few miles from Cambridge. Cambridge is there. You've got King's Lynn up here. You've got Norwich and Ipswich, and this is the area of East Anglia, this general area here. So that's where I am, and that's where I'm speaking to you from. Thank you very much for the invitation. It's a great pleasure to be able to address you and make a reach across the pond to share ideas and opinions. And I've been listening with great fascination to what Caitlin's been telling us. And certainly the message that Caitlin's been giving you is a similar one to what we are hearing over here on this side of the Atlantic. I was asked by your team, Bonnie and Kay and Alison, to speak about artificial insemination or artificial reproductive techniques for the Suffolk Punch. And I sort of threw in the question, are these the tools for moving mountains? The stork on the top of the chimney of the Swiss National Stud in Avanches is a symbol, perhaps, of old-fashioned way of thinking about distance breeding. And certainly the stories and lores of the stork always are associated with the stork bringing the baby over a greater distance. So you may believe that if you wish. I think we will apply that thought to modern artificial reproductive techniques that could be of use and hopefully will be of great use to the Suffolk breed and trying to perpetuate it. Let's first of all have a quick think about what is reproductive success and efficiency as perceived by you, the breeder, and measured by those who are involved in developing the breed. Obviously, the most important thing to the breeder is to have a live foal 11 months after the breeding event, and preferably a healthy live foal and all that entails. Now, that is a fairly crude way of measuring reproductive success, although a very effective one. What we are actually more interested in many ways, is how to assess reproductive efficiency. And for that, we can use a very simple rate of per cycle, large in effort, breeding season or breeding lifespan. And that is intricately then linked to the cost of breeding, the financial cost of breeding. So whilst the live foal rate is the ultimate outcome that we're looking for, the per cycle conception rate is the measure by which we can assess the efficiency of our breeding programs. And that we usually measure at a far earlier point in time, and that is usually at the first scan, the first ultrasound scan or pregnancy diagnosis, which is usually somewhere around 14 days post-ovulation. And here on the left, you have an embryo, embryonic vesicle, with an arrow showing here the tiny, tiny embryo, and it's about a half an inch, or the diameter of this vesicle is about a half an inch, and that we can pick up on an ultrasound scanner as a round dot. And that gives us an idea whether the breeding effort was successful, but of course it doesn't tell us whether we'll get to that stage yet, as it's still a long way to go, and all sorts of things can happen between this stage of pregnancy and the ultimate outcome of the live foal. But this is a good start. There are various natural assisted reproductive techniques, pasture with different levels of efficiency, breeding efficiency. Pasture breeding, as we can see here in this group of conic horses, are literally across the road from me in an area of about 800 acres. These are a herd of feral ponies that breed there naturally, untouched by man, and they have an extremely high efficiency, reproductive efficiency, and probably, certainly 90% of them, 9 out of 10 mares, if not more, closer to 10 out of 10 mares, are in foal at the end of each breeding season, and theirs is a completely natural breeding season. They start covering their mares as summer approaches, in late spring, roughly in April and May. Their foals are born in a roughly 60-day window, and they put us to shame 5 miles down the road in the high-tech world of thoroughbred breeding, where we achieve similar conception rates, but at a far greater cost and financial cost, because of the various intricate obstacles that our breeders put in front of themselves to want early foals. So there we have a very efficient natural way of breeding with a high fertility rate, assuming that the parent animals are fertile, and assuming also that there is some diversity and genetic diversity in that group, which is guaranteed or which is enforced by the behavioral aspects of that group of horses, and in particular the male horses that are dominated by a lead stallion and several bachelor stallions who get every now and again to nip in and have a bit of hanky-panky with one of the mares while he's otherwise engaged, and the other group are kept well away, the very young stock. The mares themselves tend to also avoid breeding directly to the most closest of their relatives, which is also a fascinating aspect to their behavior, and has in recent times been shown to, a foundation has been shown through work done by a Swiss group and a German group looking into major histocompatibility complexes and how these affect the attraction of individuals to each other, be that in horses, and also they used studies on students by using things like armpit tests and things like that, and found that sniffing somebody else's t-shirt, you might find one person more attractive than another, and that can then be linked to to blood markers as well. So food for thought there, but very interesting from a behavioral point of view. Hand breeding is what we traditionally associate with traditional breeding methods, where the mare is brought to the stallion and covered at what we the handlers and owners think is the right time for the mare. Without veterinary intervention or close management, the efficiency of hand breeding is actually quite poor. We're probably looking at no more than probably 40 to 50% per cycle, if not less, whereas intensive management, we know, has got a 70, 80% per cycle conception rate, and pasture breeding is up there at that level as well. So man's intervention and poorly done intervention is actually detrimental to breeding efficiency. Now, this is of great concern, of course, when we start introducing assisted reproductive techniques, because we want to achieve high breeding efficiency and also achieve high financial efficiency as well. But the investment in assisted reproductive techniques will cost us financially, but also will have benefits if it's done correctly to help the plight of small breeds and the aim of trying to get the male and the female to meet over great distances. And there the limiting factor, of course, is the cost and the difficulty of moving, especially the large breeds such as a Suffolk. So what are the natural and assisted reproductive techniques that we could think of using? Well, the simplest and the most efficient in the short and medium term, particularly for this breed, is artificial insemination, followed by embryo transfer, which is becoming, which has of now a very well-established place in the breeding of a number of breeds. And then we can start thinking of more advanced techniques like ICSI, intracytoplasmatic sperm injection, or even cloning, which could be of benefit if we really run into a problem with trying to find the appropriate male animals, especially to perpetuate the breed and guarantee genetic diversity. I would like to share with you a short video here, because I think this is a fantastic video, which shows what we're seeing here in pasture breeding, but at a microscopic level. And this is actually shortly after intracytoplasmatic sperm injection, the first phase in a time-lapse video from work done at Liverpool University and in conjunction with Taliesin Matson up at the Stallion AI Center. And this is a fantastic picture of, you see the first cell divisions following the introduction of a sperm cell into an oocyte, into an egg cell. Whoops, there we go. So that's the creation of a horse life, which is, you can see the cells as they are developing here, as they're splitting. This is obviously in time lapse. And here we go, division of 2, 4, 8, 16, 32, so forth. And that is the creation of a horse. So let's come back to earth a bit and talk about artificial insemination and how it can be used practically for breeding the Suffolk horse. Basically, as you probably know far better than I do, there are 3 different forms of semen that we can work with. That's the freshly collected semen, which has a shortish lifespan outside of the horse, the stallion's, and especially the mare's body. Chilled semen, which gives us a window of about 48 hours, maybe 72 hours, to work with the semen outside of the mare. And frozen semen, which, if processed correctly, or when processed correctly, gives us an unlimited time span of storage. The oldest semen that I've used from horses was over 20 years old, and in warm bloods in Switzerland many years ago. And the fascinating thing about using this very old semen was that we were from a very, very popular and well-bred stallion in its prime 20 years earlier, was that when we bred him or his semen, he was long dead, to modern sport horse mares, expecting another superstar to be born, it was quite a disappointment. Why? Because the breed had made huge progress in over 20 years. And actually what we were left with, or what the result of this old semen from this old stallion, was a retrograde step in that we went backwards to a type of horse that was popular and competitive 20 years earlier, but no longer competitive in the modern world. That's how fast the warm blood breeds have developed since the Second World War, and especially in the 1980s and 90s. The consolidation there is now rapidly progressing. So let's think about stallion reproductive physiology a bit. The testicles produce billions of sperm per day. It takes testicular tissue about 57 days to produce a sperm cell. It then leaves the testicle and moves and is stored in the epididymis for a period of up to 10 days, so that the full cycle to produce one sperm cell is roughly 62, 63 days, somewhere around there. Once it's released from the epididymis, it is not just voided through urine, but is actually used for reproductive purposes. It is mixed with secretions from the accessory sex glands further along the male reproductive tract, and that produces the semen. And you can see in the baby bottle down here, an ejaculate from an Arab stallion or an Arabian stallion, roughly 30, 40 milliliters of ejaculate, and that will contain a sperm-rich fraction and the fluids that are produced by the accessory sex glands and the seminal plasma. The picture above, you see the late Mr. Morley covering a mare at Nigel Oakley's farm, Reed Hall Farm near Bury St Edmunds. And this is probably the traditional way where the stallion in the Suffolk horse would travel to the mare. In many other breeds, it's the reverse, where the mare travels to the stallion, and the traveling stallions would appear on the farms at given intervals, but not necessarily at those that are absolutely appropriate to the mare's estrous cycle and timing for mating or covering. That nowadays, obviously, with the appropriate management, can be corrected. So let's go back to the stallion for a second. This is a table from 1949 from a German veterinarian called Dr. Goetze, who wrote a treatise and a fantastic textbook on artificial insemination in various species, including the horse, in the 1930s. It could only be published after the Second World War because of the trials and tribulations of the Second World War, but he based his work on work that went back to the turn of the century, and in fact artificial insemination was being used in the 1890s in the imperial Russian studs in Russia with great success at that time. So it's nothing new. AI with raw and also an extended semen is nothing new. Extenders are already being used based on egg yolk and milk and so forth in the 1930s, and the Hanoverian Breed Society was using AI quite extensively in those days. Well, they also bred heavy horses, or what they call draft horses, and here are some measurements or parameters of what an ejaculate looks like, and comparing those to different breeds. So the draft horse, he collected 21 stallions, 362 ejaculates, and the average volume, the total volume of semen was 145, almost 150 milliliters, so close to half a pint. But the range, as you'll see on the right-hand column, in this column here, is quite large. It goes from 35 milliliters, which you saw in that Arabian horse a minute ago, to almost 2 pints or half a liter of semen. Now, a lot of that, the variation or variability, is due to the not the semen rich fraction, but primarily due to the accessory fluids that are produced and are often quite based on due to vary because of the horse's level of excitability and so forth. You'll see the similar numbers were seen with warm blood horses. These would be warm bloods in the 1930s and 1940s, which were far heavier. These are the heavier Hanoverians, probably quite far closer in weight and in some instances in type to some of the lighter German draft horses from northern Germany. Other breeds, he had a few thoroughbreds that he looked at. They were batting at a lower average, and the ponies were batting at the lowest average. So we assess semen by looking at the volume that the stallion produces, and we divide that into the sperm-rich fraction and the gel-rich fraction. The sperm-rich fraction is what we are interested in. That contains the semen and the sperm cells, and that is what we're going to work with. From that, we want to know how many cells there are per unit of volume, so how many per milliliter. So we'll calculate the semen concentration, which will then give us the basic information to create or divide an ejaculate into doses depending on what form of AEI we're going to use, be it chilled semen or frozen semen. We will then also assess the motility as a sign of whether the semen is alive, and we will assess that usually subjectively, although there are in big centers now, this is done with assessors or motility machine analyzers, but basically it's on the day-to-day basis. We assess this by estimating what percentage of semen is moving and motile, and we assume that the semen that is not moving or motile, that is dead. And if we multiply that by the semen concentration, we will have what we believe is the amount of live semen in the ejaculate. So we'll then be able to decide how much total live sperm is available for insemination. Further tests can be done and are often done in a laboratory setting, especially if we suspect that a horse has got a problem, a fertility problem, or when we're assessing young stallions prior to their breeding careers. What's work that was done in the 1970s by Dr. Ed Squires, who is now in those days he was up in Colorado and is now at University of Kentucky at the Gluck Center, but was very basic physiological work, showed how a stallion depletes himself with multiple ejaculations during the day. And I'm just throwing this slide in so that you see what the first ejaculate of the day, the total volume is high, the 50 milliliters of semen, and that is divided into the sperm-rich fraction, which is the dark blue column, and the light blue column, which is the gel fraction, which was cast away. And you can see how the gel fraction changes quite rapidly, but the sperm-rich fraction drops less rapidly during the over a course of multiple collections, spaced roughly equally apart during the day. Now, if we translate that or look at what happens to the semen concentration on the similar sort of graph, you'll see this. Here we have high concentrations, of billions of sperm per milliliter. So first ejaculate of the day, very high. And then it drops quite dramatically. And I did this with 2 different Arabian stallions a few years ago, just as a teaching instrument to some of the staff on a stud farm. And we collected these horses also 5 times a day. And you can see here how just there's a difference in the intensity of color of the semen and how it becomes lighter or more transparent during the course of the day, and how it differs between 2 different stallions. So there are great variations within stallions and between stallions. And when we come to assessing how much of this first ejaculate is available for shipping, we could subdivide this into probably 6 to 8 mares and probably 10 mares for that particular ejaculate for that day. And this will be the case with this fellow here, but possibly not with this fellow down here. So the importance of knowing how much semen is available will have a bearing on how many doses you can prepare to ship or to mares in different destinations, or to use to inseminate on the farm. Interestingly enough, the thoroughbred world gets very, very excited because, as you know, the thoroughbreds are the last of the major breeds that do not allow any artificial or assisted reproductive techniques, and they're very keen They're very worried, breeders, quite often, to get the third cover of the day. Well, the third cover of the day is probably that one there, and that would probably still be enough to cover 5, 6 mares. And so there's plenty of semen available at that stage, and we're hitting AI levels only after the 5th or 6th ejaculate. Certainly, if we're working with frozen semen, where very small doses of semen are actually used. We need to be able to measure the semen concentration. There are various methods of doing this, be they mechanically or with a microscope. I won't go into those, but those you need to be able to assess these parameters to be able to provide a good service. This I threw in because I thought it's quite interesting. This is a stallion, an Arabian stallion, and this is the course of a whole season over 4.5 months. You can see he was collected sort of on an every other day, every third day basis, and you can see how the concentration of semen varies from day to day and across the season. And we did this with 7 stallions, and each stallion had his own sort of pattern, but the general trend of having relatively high concentrations early in the year, in February, March, and then lower concentrations as we went along, with higher volumes, shows a very similar trend or pattern. So don't expect the same level, same concentrations or same volumes on a day-to-day basis. This changes from day to day, and from the frequency of the frequency of the stallion's use. Semen motility, as I said, we look at it as raw semen, and we reassess it in its extended form, because we will add extenders to the semen to extend its longevity outside of the mare's reproductive tract and in the media that in the systems that it is being processed through, be that chilled semen or frozen semen. And this is basically an automated system that looks at a field of view and then measures various parameters, motility parameters, and gives you a reading. And it's obviously more objective than the subjective, our own personal subjective assessment of semen, which is actually quite repetitive of the same person's looking at the same sample the samples all the time. Right, factors affecting semen quality. This is extremely important with regards to providing a good product to the end user. And these are some of the important factors. Operator experience. If somebody with increasing experience in collecting the semen from the stallion, knowing your stallion, knowing when he's comfortable in an AV, that has a great bearing on the quality of semen that is collected. But far more importantly is that semen does not like being exposed to temperature fluctuations. So it's obviously produced at roughly body temperature, and then it likes to stay at body temperature, which, if you think about it, is what happens in nature. It's being ejaculated into the mare's reproductive tract at body temperature. But what it will tolerate is gradual changes that are continuous but not erratic. So it does not like, semen does not like being exposed to erratic changes of temperature, and that kills off semen very, very rapidly. So that starts with the whole setup of the artificial vagina, the water temperature inside it, and so forth, and then how it is then collected into the AV itself, the containers, the bottles that it's collected into, and then how it's handled inside the area where semen is processed. So steady temperature changes are okay, but extreme ones are not. Semen also doesn't like light, does not like being exposed to light. So it's best off from the collection receptacle going straight into a darkened area, into a dark container, be that a refrigerator or eventually then a shipping container, which is is closed off to light. It also doesn't like being exposed to oxygen. That kills off semen as well. So the quicker we can get it into its shipping containers or its holding containers that are oxygen depleted, the better. It also, strangely enough, does not like being in seminal plasma or in the gel fraction. So the quicker we get it out of the gel fraction, the better. And that, of course, if you think about the physiology of ejaculation, makes sense too, because the semen-rich fraction is ejaculated directly into the uterus, and the gel fraction is actually only produced afterwards and is left in the mare's vagina, and it itself has got cytotoxic or spermatotoxic effect, so it kills off semen if the semen's left inside the gel fraction. So we need to separate that very quickly. Right. That is basically some of the physiology and the collection process and the thoughts we have to have about how we handle semen. But we must also think about the risks of infection and venereal infections. And of course, if we ship semen from a stallion that is carrying a venereal infection, that venereal infection will also be perpetuated in either the chilled semen or, for that matter, also in frozen semen, and can be and will be spread far and wide unless we ensure and make sure that the stallion is healthy and does not carry any venereal infections, be they bacterial, viral, or fungal. So what you have here is an illustration of how stallions are swabbed prior to the breeding season. And these are requirements not just prior to the breeding season, but if you're going to collect semen for export and for freezing or for shipping, you will have to do this too. And you should be doing this if you're shipping, if you are shipping semen from your stallion to mares at other locations. This here is a bacterial infection called contagious equine metritis, caused by a bacterium called Tailorella equigenitalis. And this is a notifiable disease also in the United States, but certainly also here in Europe and most of the world. It's recognized by the OIE as a venereal pathogen and has caused great damage to breeding industries, be they thoroughbred or otherwise, in various parts of the world. So I cannot stress enough the importance of breeding shed hygiene and the hygiene of both the mare and stallion, and in particular, if we're using stallions semen for shipping, for commercial purpose and others, this has to be for perfectly healthy animals. Here's a semen collection. This is an Arabian stallion in pretty good conditions, using a dummy, and the stallion arrives into the breeding shed. He has been conditioned to recognize this beautiful mare here as his girlfriend, most stallions can be conditioned to recognize and mount a dummy. Otherwise, you sometimes just have to have a mare nearby. She might have to be in season, or maybe one that is not in season, just to stimulate the horse. And of course, the other way of doing it is by mounting a in-season mare and deviating the penis into the AV to collect semen with a stallion mounted on a mare. So here we have the collection process. In he comes, the raging bull, hops on the AV. You can see he starts thrusting, engaging, thrusting. This is a tail flag here, and he has ejaculated, and he has then semen's been collected. This is our chairman of the Suffolk Horse Society's breeding committee, John Fleming. It's several years ago, bringing a stallion into our practice, to our hospital facility, where we were collecting semen to inseminate Dorothy, one of Michael Tullemarsh's, the late Michael Tullemarsh's mares. And this is done, the second method of doing it, as I described earlier, which is by using an in-season mare to collect semen with. What are the other tools that we need? Well, we need an AV, an artificial vagina. They come in all shapes and sizes. This is a Japanese one called a Nishikawa. Mr. Nishikawa in the 1960s or 70s decided that aluminum was a great way of making an AV. This is what we call a Cambridge model, and this is probably the one that you'd be most familiar with, which is a Missouri model. Each of these have got their advantages and disadvantages. The Missouri is probably the easiest one to use. but has disadvantages in the sense that unless you can fit a disposable liner, you end up having to sterilize and disinfect this rubber liner every time after every use, because of course this is also part of the cleanliness and hygiene and prevention of infection process that you have to use clean and sterile material and disposable material, as we have here with a plastic liner after each semen collection, especially if you're working with multiple stallions. Semen processing, the collection area, which is to the left here, should be separate to the laboratory area, and there should be no direct communications for dirty feet to come trundling in and into the processing area. So this is just a window which the team that is doing the semen collections hands the collected semen in a little bottle to the lab assistant, and here she is processing the semen. First of all, filtering the semen through gauze. You can also decant it to get rid of the gel, and then we look. This is 125 milliliters from a stallion, or half a pint, or a quarter of a pint rather, and this will be filtered, and then immediately the semen extender is, after having taken samples out of it for analysis, the semen extender is added to it at roughly body temperature to maintain the temperature curve on the semen. And then once that is done, the semen is examined for motility and so forth, and the semen processing then goes on, be that into extenders for chilling or into the semen freezing process, which is more complex. Chilled semen has a life expectancy of roughly 48 hours. Some stallions will go up to 96 hours. It varies from horse to horse and from ejaculate to ejaculate. Some of the more modern extenders now are trying to extend this practical lifespan of semen for greater than 48 hours. My experience is that best success is achieved with semen samples that arrive within 24 hours or 24 to 36 hours after collection. Fresh semen, if it stands on a shelf in a laboratory without any extender, will die off after 3 or 4 hours, and extended semen at room temperature will have a lifespan of roughly 4 to 8 hours, sometimes 12 hours. It varies from horse to horse. Chilled semen can be shipped. Ideally, this is the state of the art. This is the Rolls Royce of shipping containers, the Equitainer, very expensive and used to be far more prevalent. Nowadays, I find that most semen we receive comes in a styrofoam box with a container, with the semen dose in a syringe or in a vial, and that is kept at a cool temperature with ice packs. That works quite well. Certainly in our more temperate climates here in Europe, in Central Northern Europe, you might find that these cool packs actually warm up very quickly in warm climates, like in the southern parts of the States, or probably where you are right now. And their temperature curves are not as guaranteed as they are certainly in these Equitainers. But this is really a question of cost and retrieval of the Equitainer. If it's been shipped halfway around the world or halfway across the country, to retrieve them is often a big struggle for the owners. Frozen semen, roughly 50%, maybe nowadays with modern extenders, up to 60% of stallions are freezable. Not all stallion semen will freeze, which is no reflection on the fertility of the horse in a natural cover situation or with chilled semen. Because the horse is not selected for freezability, as for instance beef cattle are, or beef bulls, we will always have this. We will always have a largest proportion of horses whose semen will not be freezable. But as I've said, with modern more complex semen diluents or extenders, we are slowly improving some of these things, some of these rates here, freezability rates. The Suffolk is quite interesting in that the Suffolk breed appears to pose some challenges for freezing, although those that we have frozen here, both at the practice and especially Tullus Matson at Stallion AI Services, and before him, Martin Boyle, who was the first to freeze Suffolk stallions when he was working at the Equine Fertility Unit and subsequently with Stallion Reproduction Services. We have always found that the Suffolk produces relatively small volumes of semen, and whilst it freezes relatively well, when we look at it post-thaw, on some horses, the fertility is there, but the post-thaw motility doesn't look as good as we would expect. But Suffolk can be frozen. I haven't yet been able to identify stallion lines that freeze better than others, but I'm sure they're there because I've seen that in other breeds. So that's basically having a gallop through the stallion. Let's look at mare AI from the mare's side of perspective. How do we manage our mares for artificial insemination? And again, I'm going to start off with hygiene, 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 hygiene. And I am an absolute stickler for hygiene because I can assure you that the best contraceptive is a bacterium in a uterus. You will not get a mare in foal if she has got an infection. So they have to be, mares have to be screened for gynecological health, and that is done by getting a swab or a smear for cytology or for bacteriology when the mare is in season at the start of the breeding season, and preferably with each breeding cycle if she does not go in foal immediately. So to be able to breed the mare, she has to be in season. Surprise, surprise. I've never got a mare in foal that wasn't in season. So they need to be in season. They need to be close to ovulation when you inseminate them. In fact, if you're using chilled semen, you can be within 12, 24 hours of ovulation, quite happily. But with frozen semen, you need to be within about 6 hours of ovulation to achieve good per cycle conception rates. And it's important to check the mare for ovulation to make sure she has ovulated after she's been inseminated, and far more importantly at that stage, to ensure that she that her post-AI uterine health is good. In other words, that she is not coming up with a severe post-insemination inflammatory reaction or infection. If she is, you've got time, 3 or 4 days, in which to treat the mare and clean up her uterus before the fertilized egg arrives in the uterus. So these are not good signs. This is not a good idea to breed to a mare like this. She's got a vaginal discharge, which is more than very likely, or could very likely be a acute endometritis. You can see there is a discharge running down between her thighs as well, and she is likely to be a subfertile mare. So the mare needs to be clean to be bred. The mare cycle, if we put a speculum in through the mare's vulva into the vagina and look at the cervix, this is a cervix of a mare that is not in season. So it looks like a rose bud. It is tightly shut, and it tells us that she is either not in season, in diestrus, or perhaps even pregnant. This here is a wilted rose, so the mare is in season, the cervix is completely relaxed and able to dilate when the stallion covers her and ejaculates through the cervix into the uterus. This is the only domestic breed where the semen is deposited through the cervix into the uterus, whereas if you think about cattle or pigs, it's either in front of the cervix or into the cervix itself. So this cervix is able to dilate quite great extremes to allow semen into the body of the uterus. And we have the T-shaped uterine horns here. We can see the fallopian tube here. We can see the ovary at the end of the fallopian tube and the fimbria that fit over the ovary. The eggs are produced in the ovary, released into this structure, this trumpet-like structure, and the egg then goes into the fallopian tube, where the sperm cell has attached itself to the wall and is waiting to ambush the egg. So semen's deposited into the uterus, and within 20 minutes, the semen appears in the fetus of them, of course, appear in the fallopian tube. If the lucky sperm cell manages to find a lucky oocyte or egg cell and manages to fertilize it, the fertilized egg, the embryo, will sit in here for up to 4.5, 5 days before appearing in the uterus. Now, if you think about it, the stallion has deposited the semen directly into the uterus, so he's gone through the cervix, or the ejaculation has inseminated through the cervix. We are doing that with a catheter and with semen. So there is an introduction of contamination into the uterus. Nature has designed the system in the horse in such a way that the mare now, with her immune system, if it's a healthy immune system and it's not overcome by infection, is able to get rid of this contamination back out through the cervix during the 4 or 5 days before the embryo arrives in the uterus and says, I am here, and starts moving around quite actively, again, something's peculiar to the horse or to equids, to tell the uterine wall, I am here, don't get rid of me, I am pregnant. And this is that window of opportunity where we can help with mares that have difficulties getting rid of infection or this natural, normal challenge to their local immune system before the embryo appears. So that's the rationale to post-covering treatments, post-covering lavages, and so forth. We are not damaging the embryo because it is happily still sitting in the fallopian tube, certainly for 4 to 5 days until the cervix tightens up completely and we have a clean, sterile environment. So what gets the mare in season? Well, there are environmental factors. Number one is light. The mare, as we all know, generally does not cycle regularly in winter. The further away you are from the equator, the more pronounced this winter anestrus, this non-cycling period is. And the closer you are to the equator, the less pronounced it is. Equids on the equator cycle regularly, like clockwork, every 21 days, or 21 to 24 days throughout the year. The closer you get to the North and South Pole, the sooner they switch off and the shorter their breeding period. And this is all designed to make sure that these little fellows are born in April, May, June, into the spring, when lush grass is starting to grow. The mare is in a lean condition and is then into this new fresh green grass, which then enhances her milk production to the benefit of the foal. So body condition of the mare has got a great bearing on fertility as well. And all the Suffolk breeders around here don't fear me, but they all are fed up with me saying, your mares are fat. And they are fat. Most of them are obese. And that has got a detrimental effect on fertility. Why are they obese? Why? Because they've become glorified lawnmowers and are not working as hard as your Suffolks over there that are in the timber industry. Big round contours are not conducive to efficient fertility. So nature's cycle is that at the end of winter, the mare is lean, mean, and ready to go. The pregnant mare is also lean, mean, looking for grass to then be able to nourish its newborn foal that is born into the green grass. Temperature has a certain amount of effect on fertility, but to a far lesser degree than certainly light and photoperiod and body condition. The estrus cycle is about 23 days, 21 to 23 days when they're cycling regularly, and it lasts, they're in season for 5 to 7 days, and ovulations at the end of estrus, on the 7th day, and that's when really we want to have semen available in the fallopian tube for fertilization. And as I've said, with chilled semen, you've got a far longer window. You've got 24, 48 hours from the moment of insemination to ovulation. With frozen semen, you want to be within 6 hours of that ovulation. So the interestrus period or the diestrus period is about 18 days when they're cycling regularly. And I'm not going to go into the details of the hormones, but that basically is how it works. So here's a mare flat out in season, winking away, ready to be showing us that she's in season. She needs to be followed and monitored. And the closer we get to the end of the cycle, the closer we get to ovulation, will be the time when we will inseminate the mare. We can monitor that by following her with ultrasound and monitoring her follicles. This is a large follicle that's coming close to ovulation, another day, 24 hours, 48 hours away. And we can look at the patterns of the uterus, the fluid patterns in the walls of the uterus that can be seen on ultrasound as well, which we can confirm with rectal palpation by feeling the uterus and feeling the follicles as well. So predicting the exact moment of ovulation is more an art form than a science, but it comes with experience, and we can help it along with the more modern inducers of ovulation, such as ovuplants. You might be familiar with ovuplants, or luteinizing hormone, or deslorelin, the injectable form, and so forth. So, horses being horses and mares being female, they have their peculiarities, and they have the right to have peculiarities as well, and they've got abnormalities of the cycle. What is peculiar to the horse is the foal heat. At 8-9 days post-foaling, mares will come into season, and quite often this foal heat is used for breeding. We do not tend to use it here, and I certainly don't use it regularly for AI. AI in mares, I tend to go to the second heat period, or try to induce a heat period about 8-9 days, a week after the end of foal heat. The foal heat to me is a cleansing cycle for the mare. If we look at the stallions down on Wick and Fen across the road here, who are not touched, or where the horses are not managed, very few of them will actually cover the foal heat mares, which is an interesting observation, which is not necessarily the same in other feral herds. Mares can be very annoying, especially if you want to have early foals, which I don't see any reason to have early foals, but the thoroughbred world certainly wants everything to be born on the 1st of January. And at one stage, some of our Suffolk breeders wanted to have early foals, or want to have early foals because of the showing season. But mares can have a persistent winter anestrus, which is very difficult to get them out of, and some of you will be familiar with that. The other thing is, some mares, which get the reputation of being every other year mares, have something called a lactation anestrus. So whilst they go through their foal heat, they then switch off with a foal at foot, and are also difficult to then stimulate to come into season. The other extreme, which is more of an issue in working horses or mares that are not being used for breeding, is its persistent and annoying estrus behavior during the summer months. So what is the best time for AI? Well, we want to have a relaxed cervix. Here we've got the will to the rose. We want a softening ovarian follicle. This follicle here is large and softening. And we want the characteristics of the wall of the uterus to be such that we know that we're at well into the estrus period. We need to have a clean mare, a clean bacteriology or cytology result, and we can then induce ovulation, order semen. If it's chilled semen, we'll give the mare the ovulatory agent, be it LH or Coralon or Deslorelin or Ovuplant, whichever one your vet prefers to use. And at that point, after having communicated with the stallion owner, you then, well beforehand, so that he knows, the stallion semen providers know that there's a mare on the books, the call is put in to order the semen. The semen is then delivered the next day, and the mare is inseminated. That's the ideal way of doing it with chilled semen. And you will then go back to the mare 48 hours later, check her for ovulation and check her for any abnormalities in the uterus, such as fluid, abnormal amounts of fluid, which are signs of an inflammatory reaction, and that then needs to be treated. This slide is about 30 years old, no, 25, not 30 years old, I guess. Nothing's really changed, although this ultrasound machine went out the window many years ago. But this is the gear, the basic gear that you need for chilled semen insemination: disposable gloves for examination, disposable gel, cleaning material, disposable water in a disposable container or liner to be able to wash the mare, clean her up, and then inseminate her. Frozen semen, more complex, more complicated. The frozen semen process is a more complex one. Stallions usually have to go away to collection centers for freezing, and they definitely have to go away to collection centers for freezing if the semen's going to be frozen for export. This is an expensive process, the export process, because you have to decide where your export market is and freeze the semen to the health requirements of the country of destination. The United States is particularly complex in its requirements, as is Australia and New Zealand, and those are the markets, the primary markets potentially for Suffolk stallion semen in the UK. We have sent semen quite successfully. I've exported it out to Australia and New Zealand, and the semen, some semen that was frozen in the course of this last year has been frozen to North American import requirement conditions. But it adds a substantial amount of cost to the freezing process, and obviously for the end user to the end user's costs as well. The important thing is to realize that there are different types of freezing methods. You will probably nowadays just find semen being frozen in these little 0.5 milliliter paillettes. Usually, I say usually because it varies, 8 straws are 1 dose. This is an old-fashioned macro tube, which you'll probably not see, I almost certainly will not see for in Suffolk horses. This is still used, some of these are still around from warm blood stallions and so on from Germany. The important thing to realize with frozen semen is the timing is more difficult, and it is managing the mare is more complex, and with that comes all the additional costs come up in veterinary fees and management fees. Ideally, frozen semen is used in the mare is brought to an insemination station for that. I've been inseminating mares on the farm, on owners' farms now more recently, using fixed time insemination rather than having to bring them into the clinic to do it there, and that has worked quite well. You need some minimum facilities on a farm to restrain the mares and so on, so that you can work in good conditions. So fixed time insemination, the stopwatch is not Big Ben, but we could use it as that. Basically, we have the same criteria. The mare has a relaxed cervix, the uterine wall pattern is advancing, and the follicular diameter is over 30 millimeters, or in other words, about half an inch. In the Suffolk, I find Suffolk follicles are maybe a bit larger than some of the warm blood and thoroughbred mares at the time of ovulation, but not significant, not as a general rule. What I will do at this point, usually at 8 o'clock in the evening, I will inject the mare with something called deslorelin, which induces ovulation roughly 36 to 40 hours after injection. So we have injected the mare at 8 in the evening. I might go back the next day, 24 hours later, just to assuage my own conscience, check the mare late afternoon, and then go back the following morning, so that's 36 hours later. And I've, in a group of, I think it was 15 mares this year, Arabian mares, we had ovulation in 12 of those 15 between 8 o'clock in the morning and roughly 1 o'clock in the afternoon. So we could happily inseminate early in the morning, or at 8 o'clock in the morning, and we knew we were within we would be within, or the chances were we would be within the 6 hours was very high, and we had a 90%, or 9 out of, what was it, 12, we had 12, I think 10 of those 12 mares went in full on the first cycle. 2 Suffolk mares that I inseminated that with the same protocol this year went in full on the first cycle. There are other protocols that you can do as well. You can also just basically follow the mare every 6 hours once you've injected her until she ovulates, and then inseminate her. Those are all perfectly legitimate ways of doing it, but this fixed time insemination has worked quite well this season. Probably be different next season, but it seems to be a fairly good protocol and has worked with Suffolks, and I did not have to move the Suffolks from their farms to the clinic to do that. What's important is the following the mares after the ovulation. So as I said earlier on, we check them for ovulation and see whether there's any unwanted fluid in the uterus, at which point we treat them by using something that will help squeeze the uterine wall to help squeeze out this fluid, something called oxytocin, which is the same hormone that is responsible for milk letdown, which is why I'm so keen that all foals and their mares are turned out into paddocks, small paddocks, immediately after foaling, especially early in the year, to make sure that the mares are moving and that the foals are on suck. And with that, the uterus is encouraged to contract, and all the postpartum, the post-foaling fluids and junk gets pushed out as well. So oxytocin is used exactly in the same way after covering, and you can use that quite comfortably after ovulation for a certain 2 days or so, along with flushing. And when in doubt, flush using a saline flush. Here's a 3-liter bag, and you can see this one is cloudy and murky, and that was immediately after, a day after an insemination with chilled semen. And we'll flush until we get a clear flush, and when in doubt, flush. We're using less and less antibiotics. I don't like using antibiotics. Most mares don't need antibiotics post-covering. Flushing is enough. They just need help to get rid of the junk inside their uterus. But if I do, I will do it. I try to only use antibiotics if I know what bugs are being grown and what their sensitivities are. There is an idea, if you have got infections, post-covering infections with streptococcus bacteria, that a course of systemic potentiated sulfonamides or trimeds can be of benefit. Other things to treat a uterus with are plasma, or what's becoming more and more fashionable is use of platelet-rich plasma, which is basically what used to be plasma in the old days in a more concentrated form. But we are consciously trying to get away from the use of antibiotics unless absolutely necessary in mares, both on the thoroughbred front and across all the breeds. So what are the pros and cons of AI? Well, the pros are the cost, and the cost in relation to, with regard to the Suffolk, with the cost with regards to moving animals and distances. So you would reinvest a fraction, I would have suggest, over time of your shipping costs, of movement costs of the Suffolk Mountains by using AI. The other advantage is hygiene. It stops the spread of venereal disease if the donor animals are clean. There's a reduce of injury at the time of covering. Stallion management can be improved and done locally, and mare management is improved as well because of the intervention, the necessary intervention with veterinary assistance. On the converse side, costs are also obviously a downside, but I would argue that especially with Suffolks and having to move big horses like that, that over time these costs the advantages outweigh the disadvantages. One of the big issues as a complication, especially with chilled semen, are shipping complications due to couriers not getting from A to B on time. In other words, semen being lost somewhere in the courier system, if FedEx or whoever they are shipping it, or you're shipping during the spring holiday season over Easter. That is a hopeless time to ship semen in Europe, especially if you're going across borders, and causes a lot of angst because the semen is not arriving on time. Obviously, if a stallion is dirty, is infected with a venereal pathogen, with a venereal infection, the spread of venereal disease is accelerated and geographically over a wide area. So that is irresponsible. The thoroughbred industry particularly will not allow AI because of fears of overproduction, which is a false fear because they're overproducing anyhow. I don't think we have that big an issue. That is certainly not the issue that we have with the Suffolk horse. So overproduction is not an issue here. Injury risk, occasionally you still can have injuries with horses falling off dummies or having an injury at the time of collection, but I think those are certainly far fewer than the risk of injuries in natural cover situations. The other advantage, of course, for the Suffolk and for rare breeds is being able to move semen from one focus or one area of genetic density to another area of genetic density, and thereby judiciously exchange genetic material and encourage diversity of the breed. And that should not just be nationally or locally, that we should be encouraging each other to do that across populations, in particular the North American and the British population. And I'm a firm advocate of breaking down the barriers and getting into a situation where we can happily and confidently exchange semen across the Atlantic, as we are already doing with very small populations in Australia and New Zealand. So I have bored you to death, I'm sure, and I would just like to thank you for having given me the opportunity to speak to you, and I hope that I've been able to help sort of give you ideas and food for thought. I'm very keen that we should explore all possibilities to encourage the conservation of this breed, and that we should all be able to collaborate and work together across the breed registries, etc., and help conserve, preserve, and allow this breed to develop, in whichever way and into whichever markets that you, the breeders, can identify to do this in. So thank you very much for your attention. I'm quite happy if the system allows us to take some questions, and then I'll go to bed soon.
All right, Ali set me up for my section here. Folks, I'm pretty brain dead. It's been a couple of long days and actually a whole long week, and I know everybody's in the same boat. So we're going to make this happen very quickly. If you need a break, take it, but I need your attention. I'm ex-Navy. Y'all can hear me, can't you? Okay? So I will wake you up. And we will all be part of this conversation. This is so very important. Get my PowerPoint up here. That was amazing. I have never known that there was that much to know about it. But and some of y'all.
Does anybody have any questions for Dr.
Is he still waiting?
He's still here. You're still with us, right?
I'm still here. I'm still with you. I've just I'm taking my bedtime glass of red wine, and I'm going to disappear very shortly. It's 2:30 in the morning here.
I was like,
let's do that later. If somebody has some, we can work questions to him and get them out to everybody. We can do that.
Does anybody have any questions for him so he can go to bed?
Thank you. Thank you. Night night. God bless.
Thank you very much.
Thanks, turning on mine.
I appreciate it.
All right, good night. Thank you.
Thank you, sir.
Thank you very much, and look forward to meeting you all at some point. Sometime I'll be in Kentucky in a couple of weeks' time for the Breeders' Cup and the Thoroughbred sales, and I had a great time down in Tryon got out just before Florence hit a few weeks ago.
Thank goodness.